So that really allowed us to start looking at phenotyping the different plants, such as, for example, looking at area of the individual plants, but also growth patterns, plant condition, and the canopy cover of those plants. So here's some examples of that. We also looked at yield and biomass before harvest. So those results were published a few months ago. Those results were published a few months ago in the Frontiers in Artificial Intelligence journal. And those results showed that basically we could estimate, using a random forest-based modeling approach, we could estimate yield and biomass at harvest up to 4 weeks before harvest. The results also showed that to properly map the salt irrigated plants, we had to have or use a separate random forest model for those plants. So as I mentioned, we collected a lot of thermal data sets. So to be able to use them properly, we developed a radiometric calibration approach. So those results have just been published in the Census Journal a few weeks ago. So this approach, basically the whole setup was a lab-based setup to develop an ambient temperature dependent calibration function for calibrating the thermal data. So initially we calibrated some resistance temperature detectors, which we then attached to a black body to measure temperatures of the black body from 10 up to 55 degrees. So the black body was first heated up to 55 degrees and then just left within an environmental chamber at a set ambient temperature, also including our UAV thermal sensor, which then measured the gradual temperature decrease of the black body as it cooled down. And we did that at 4 different ambient temperatures, basically to develop a calibration function for each individual pixel of the camera. So what we ended up with was a calibration matrix we could apply to all the thermal photos. So what you can see here in the two graphs at the top, you can see quite a significant reduction in bias and the Rubin square error between the actual black body temperatures and the measured temperatures. But what you can also see further below is that the vigneting effect was significantly reduced once applying that calibration method. So to actually test how that calibration method worked in the field, you can see here some ortho photos along a flight line at the top. That's the standard deviation before we applied the calibration approach. And at the bottom, that's the standard deviation after we applied the calibration approach. So you can see quite a significant reduction in the standard deviation. But to take that a bit further, we also looked at wind effects in relation to flight direction. So you can see here, that's all the flight lines on the left from the outbound flights. And then we have all the flight lines from the UAV flying in the opposite direction. So what you can see here is that if you actually subtract those two, you will see temperature differences up to at least 2 degrees difference, depending on flight direction. What you'll also notice, you can actually see if you start from the top and then look further down towards the bottom of these thermal plots, you can see there's a slight increase in temperature, and that was because the temperature basically increased from we started flying at 9:15 until we finished at 9:30. So we are currently working on developing a new kind of orthomosaic approach that also take wind correction into account, just to improve on some of those currently existing orthomosaicing and blending methods that's around. So basically developing an approach that's a bit more suitable for thermal image data. So applying all those corrections, at the moment we have some other research going where we look at mapping temperature variations of plants and relating that to salt stress within the tomato fields. So we have been assessing both, like what are the results like if you just look at the delineation based on the multispectral data? What are they like if you take out, for example, some of the edge pixels that may represent mixed pixels of sandy background? What happens if you take out senescent vegetation within the plants? And what happens if you only look at sunlit part of the plants? So basically taking out all the shaded vegetation. So that's research currently in progress. But if you look at this graph here, which represents just the sunlit parts of the plants, you can see that's the difference between the plant temperature and the ambient temperature. And you can see so far it appears to be higher for the salt irrigated plants throughout the growing season. We're also doing some other research which is ongoing using the hyperspectral UAV data that we collected. So one study is looking at various vegetation indices and relating those to chlorophyll and other kind of plant pigments that were derived from leaf samples in a lab. And we also have another study that's looking at mapping the timing and extent of flowering. So you can see those little yellow flowers in the photo at the bottom, and also mapping the presence of tomato plants. So, of the tomato fruits. So if you look at this graph here, you can see the purple line, that's the flowers. So they are kind of somewhat distinct in the green part of the spectrum. There's one characteristic that was quite unique for the tomatoes. There was quite a clear trough just before 900 nanometers. So we're using a lot of that information to develop some spectral unmixing approaches to map flowering and timing of flowering, as well as the presence of fruit. Okay, so in another study, we're looking at discriminating infested and healthy date palms. So in Saudi Arabia, there's quite a problem with red palm weevils. Red palm weevils, that's that little red beetle you can see in the photo. They generally lay about 200 to 300 eggs that turns into those grubs that then live and eat the interior of the date palms until that date palm becomes completely hollow inside and basically falls over when it gets windy. So far we have used multispectral data, thermal data, and hyperspectral data to map date palms and see if we can discriminate healthy from infested trees. And to be honest, we haven't had much luck at all so far using any of those sensors. So to investigate a bit further, in early February, we installed a number of apogee thermal sensors on top of the center of a number of date palms that were both healthy and infested. And what our initial results showed now, so basically they're recording every minute the temperature, and they have done that for almost 4 months now. So basically it shows that we're basically flying at the wrong time. We're flying generally with the thermal sensor between 12 and 2 o'clock in the afternoon, whereas the main difference really in temperature between healthy and infested date palms seems to be in the morning around 8 o'clock and in the afternoon around 4 o'clock. So once we are actually able to go out in the field again, hopefully in the near future, then that's definitely the times that we'll be targeting to fly our thermal sensor. Okay, just another plant trial study that we are a part of. So that's led by another plant science group at KAUST that's looking at identifying candidate genes for the domestication of related traits of millet. So millet is a grain that's quite nutritious and eaten a lot in the western parts of Africa. So where the plant science group is looking at the genetic analysis, we are looking at phenotyping the plants, looking at plot height, plot area, growth patterns, maturation time, biomass, grain mass, and lodging effects as well. Yet another plant trial study that we're looking at that's focused on phenotyping quinoa varieties. So that's another grain that you might be familiar with. So that plant trial ran over winter, and we have a repeat plant trial running here this coming winter as well. So that consisted of 756 plots, each of 1 by 1 meter in dimensions, and within each plot there were approximately between 30 to 40 different quinoa plants. We had 141 different accessions, and again, 4 of those plots they were irrigated with salt water, and the other 4 plots they were irrigated with fresh water. So we had a total of 7 field campaigns during the growing season, where we collected field data as well as RGB, multispectral, hyperspectral, and thermal imagery. So the results are still pretty preliminary, but we had some pretty good results so far using all the flying dates and looking at LAI, or leaf area index, and chlorophyll, with very nice correlations with field measurements and those derived using a random forest modeling approach from the UAV data. Also, plant height using regression analysis is looking pretty good. So one thing we did find though was that there was actually very, very small variation between the accessions of the quinoa plants grown under salt irrigation and under control conditions. And that's basically because quinoa is naturally quite a salt tolerant plant. Some types of quinoa can actually grow in brackish water. But there's very little research out there showing if which accessions can actually grow in brackish water and how they perform in terms of yields. So we'll be looking at yield too, but we're still waiting for those results to come in. But if you look at, for example, chlorophyll, you can see how that decreases during the growing season and then decreases again towards harvest, especially for the salt irrigated plants. So you can see some minor subtle differences between the salt and control plants, but they're not that distinct. So at the moment, we are also developing a number of stress tolerance indices. So one you can see here on the right-hand side, which shows there are in fact variations in the salt tolerance and stress tolerance between the different accessions. Okay, so just the last slide or two here. So we've done some initial monitoring of olive trees up in the world's largest olive plantation in northern Saudi Arabia. That plantation has about 10 million olive trees. So we have installed 2 eco-variance towers, a climate station, as well as sap flow meters, which so far, based on thermal, UAV data, and multispectral data analysis, has enabled us to map transpiration of trees, but also evaporation of the area. That work, however, is to be quite heavily extended later in the year, where we're starting a 3-year project, where we will be looking at not just olive trees, but also other tree crops such as apricot, plums, apples, and peach trees as well. And we'll explore some kind of new technologies that are not that often applied, actually, especially in orchards. So, for example, we'll be installing a cosmic ray nutrient sensor and also some global navigation satellite system reflectometry receivers. So these they're basically taking advantage of signals that's already there. But by looking at the difference in reflected signals and those incoming signals, you can start looking at not just soil moisture, but also vegetation water content and also biomass. So we'll be exploring those together with a whole range of other field-based measurements to see if we can map all of these parameters, including vegetation structure and tree yield, and both yield quality and quantity from UAV data. And we're also planning to develop an approach that can scale our UAV data up to planet CubeSat type satellite image data. So that's pretty much an overview of the current kind of UAV-based activities that we are currently undertaking at KAUST. So a fair bit going on in that area. But at the moment, we certainly had the time to do all the analysis, as there are no distractions with extra field work. So that's it for me. So any kind of questions?
Thank you very much, Kasper. Very nice presentation.
Thank you.
Very nice. A lot of activities. I have a comment because when you shown at the beginning of your presentation the vignetting problem of thermal data, within the other working group, the one of soil moisture monitoring, we had 2 acquisitions with thermal images. One was at sunrise, and the other one was at noon. And we noticed that in the at sunrise there was like a trend within each thermograph of temperature, while this trend was not present during the daytime acquisition with the same sensor. So there is something that is similar to the vignetting effect, but it was strange that in our case, this trend was evident only during the nighttime thermal acquisition. So I'm wondering if the vignetting problem is an additive effect or is a multiplicative effect of the signal.
Yeah, so I guess that is a good question. There could be a lot of different things happening. I guess wind effects is definitely one of those parameters that's really affects your results. So I don't know the ambient temperature, if that could have an effect, but we definitely identified ambient temperature as a potential issue. So that was why our calibration approach was it was an ambient temperature dependent calibration approach for those vigneting effects. and that really helps using one ambient temperature with multiple. So it's hard to say before you do a bit more analysis of the data and look at what it could be, but yeah, working with thermal UAV data is certainly not an exact science. It's not easy.
Yeah, actually, we tried to relate this effect to the wind conditions. Okay, and it was strange that when the flight changed direction, this trend wasn't changed accordingly, but it was always in the same geographic site of each thermal image, even though the flight changed its direction of acquisition. So maybe something that is more related to the wind or some environmental condition than vignetting.
Yeah, could be. Yeah. So if you want to talk more about it at a later stage, we can certainly do that. And we have a few people that's quite specialized in the analysis of thermal data, so maybe we could include them as well.
I have also another question. Or there are some questions from the audience. No?
Yeah. This is Felix. You
Felix.
mentioned,
Okay.
Next presentation, you mentioned scaling with proximal, UAV, and satellite. Can you give more information? Because you just mentioned the scaling problem and challenge, actually, more than problem.
Yeah, so it's still work to be done, but what we plan to do is to use an existing approach we have developed to scale between planets, CubeSat data, and Landsat and Sentinel-2 data. So it basically takes the radiometric properties of other satellite data and basically apply that to the UAV data, or vice versa for that matter. It's up to you how you design it. So, but it's basically a scaling approach that allows to match up the radiometric properties of two different data sets. So that's what we'll be experimenting with. And once that's done, then it becomes a lot easier to actually start mapping different parameters and relate them to between the UAV and the satellite type data sets.
Okay.
Thank you.
Thank you. I have another question about the acquisition on the palms. Does the shape of the palm influence your optical and thermal data on your images? Or is it something that is related only to the functioning of the palm? Or the geometry of the palm has an effect on these images?
Yeah, absolutely. It's not easy mapping date palms because of the leaves and the structure of the leaves, because there's obviously a lot of kind of gaps between the little leaflets. So especially along the edges, you actually get a lot of background effects in your spectral signature, for example. Also your thermal signatures. So I think that's probably the main issue why we haven't been able to use both multispectral and hyperspectral image data successfully. There was simply too much variation between even healthy black palms and also the infected one. There was too much overlap in the spectral signatures because of that variation, probably because of the actual leaf structure. So that's definitely a huge problem. So mainly what we are looking at is the center part of the palm. So you start reducing the effects. The canopy is denser within the center of the date palm. So we're focusing on that area instead of the edges, because that's the problem because of the leaf structure.
Okay, thank you very much. So if there is no other question, I move on to the next presentation. Okay, thank you, Kasper. Thank you again. So next presentation is from Maria Tsiafouli from Aristotle University of Greece. Maria is... hello, Maria. You have to switch on your microphone. Maria?
Yeah, hello, hello, I'm here.
Okay, thank you.
Yes, I'm trying to put on my presentation.
Right. So Maria is going to give a speech on creating maps of soil ecological functioning using UAV data.
Okay. Do you see my screen?
Yes.
Okay. So I'm starting from the beginning. Okay, so hi everybody, I hope you're okay. So before I start with the results, this is an ongoing study. It's about using UAV data and getting smarter about soil ecological functioning. This collaboration has started within the KAUST action. In the training course in Iceland in 2018, there were Anastasia, a PhD student of our laboratory. She took part in this training course and she used the UAV data. But together with Brindis, who is working in the soil conservation service, she also took soil samples and shipped them to Greece, and we analyzed them in our laboratory. So what was our idea behind this? The idea was that with the UAV, you have the above ground part of the vegetation, but you have a hidden part of the vegetation within the soil. And there are many ecological functioning taking place. So what can we use the UAV data for getting smarter about our soil? Why would we get smarter about our soil? Because soil provides us a lot of functions, like nutrient recycling, carbon sequestration, and it basically allows us to live in the earth. But when we are looking closer, who is providing those functions? We end up with little tiny organisms that live in the soil and provide us all the ecological functions. So we need to know what is happening with all the biological diversity in the soil. So as the soil provides us our food, and as there is an increase of needs, we intensify our land use, and this leads to the degradation of soils. And here you can see in the map of Europe that in red, you have already many degraded areas that have lost significant parts of soil biodiversity and soil biological functions. So there is a need to assess soil functioning based on soil organisms and use them as biological indicators if you want to properly monitor, conserve, and restore ecosystems. So I'm sure if you are hearing me, can you give me a feedback? Can you hear me?
Yes, we can hear you.
Okay. Thank you.
No problem.
Okay.
Clearly.
Okay. So when studying soil organisms, we have a problem of scale. As you can see in the left figure, we are using a small amount of soil, and we have a month of work to find all the organisms living there. But we need a bigger scale of if we want to manage properly our ecosystems. So we need to bridge this gap. And here is where the UAV comes, because UAVs actually capturing images in a higher scale than soil samples, but still this scale is small enough to allow us to link soil biodiversity to the UAV data. So the idea is how to use UAV parameters and link them to biological parameters that we measure in soil in order to upscale what we see by the soil organisms and make maps of soil ecological functioning. So here's the first questions. Which parameters measured by UAVs are actually linked or can be linked to soil biocommunities? And this is what I'm going to show here. So this is the study area. This is in Reykjavik, Iceland. Many of you have been there in the summer 2018. In this area, which is a heathland, you can discern 3 different microhabitats. You can see the lupine microhabitat, which is an invasive species, which is actually nitrogen fixing species. You can see eroded bare soil areas, but also you can see what would be expected to be found naturally there, the moss microhabitat. So the methodology actually had 2 parts. The first part was the capturing of RGB and multispectral images by multi-rotor UAS, which was done during the training course. And we then calculated the various indices by this data. And the second part was the collection of soil samples at those 3 different microhabitats. And the analysis of these soil samples was the extraction of soil animals, soil nematodes specifically, and the analysis of this community in terms of abundance, trophic, and taxonomic diversity. We was, of course, not able to analyze all soil organisms. We were focused on soil nematodes because the most preferred bioindicators of soil conditioning and functioning in soil ecosystems. So a few results. In this figure, you can see how the values of the NDVI index are varying per microhabitat. In this, you have the bare soil, which of course has the lowest values, the moss habitat, and the lupine habitat. And this variation is significantly. And when you go to the nematodes now, you have somehow similar picture. You have also an increase from the bare soil to the mosses and the highest abundance of nematodes here in the lupine microhabitat. So we see that the NDVI index values and the nematode abundance values somehow are going together. Of course, we tested several parameters in our data set. Our data set has over 30 parameters describing the nematode community. I cannot go into detail right now. And around 15 parameters, which was calculated from the UAV data. And you can see here, take a glimpse only of the correlations. Basically, we found that the NDVI index worked best, which is actually somehow expected. It is described in a good way the details of the vegetation, the changes in the vegetation. So based on these correlations and on the ranges of values, we made a map of nematode abundances. In the left, you can see the map of the NDVI, and with the yellow, you can see all the sampling spots where we took actually the soil samples. And before this work, we actually knew only the abundances in these spots. By using this, the UAV data, we now provide a map where we have calculated the ranges actually of nematode abundances in the entire area. And you can see here the black parts. This is basically the spots where with the lowest NDVI values, but with a very high variation of nematode abundances. So in this part, we cannot guess really what the abundances of nematode are. But in the red, blue, and in the green areas, we have now a range of nematode abundance, and we have predicted in areas where we didn't sample the abundance of nematodes. This is actually a huge step because this has never done before. And in the scales of management and conservation, this is very important. Of course, our study has not ended here. I only want to mention that in a very recent study in Nature, a similar approach was used to somehow guess the global abundance of nematodes. They also used the NDVI index for modeling and mapping. But if you use, they used the data from satellites, of course. But if you use UAV data, you have a much more detailed capturing of vegetation, of course. So this is an ongoing study, and we want, of course, to improve the prediction models, and we want to create maps of other nematode parameters that are allowing us to better assess the soil ecological status. And here is an open request. Who is interested would like to join this manuscript, please send me an email, and I will put you in the group. Thank you very much.
Thank you, Maria. Thank you. Any question? I would like to ask if which is the effect of the spatial resolution on the quantification of your maps. So is something just about the detail of the map, or the spatial resolution in some way influence your results?
Yes, the spatial resolution influences the results because you have a much more much more detail of the indices.
So it's just about the detail.
Yes, yes, it's the detail which you have by UAVs and not by satellite data.
For example, with the lower spatial resolution, the maximum values in NDVI tends to decrease, and the minimum values of NDVI tend to increase. So with the lower the lower spatial resolution, the less the variability of the NDVI. So
yeah, I'm interested in the variation of the NDVI.
Okay. Other questions?
I would like to, yeah, I can, if I can. So the figure about the community parameters and the NDVI and other variables that you measured, it was very difficult to understand. But is there something that you can say about species composition or modifications in the community itself, or just like it's very sensible that when you have more vegetation, you will have more soil organisms. But if you can make something more knowledgeable about that.
Yeah, I can show you. Just I thought I don't have the time to show this. But I can show you an example. Just okay, just a moment. So this is the trophic structure. Do you see it?
Yeah, no.
Yeah, okay. This is not just the abundance, but this is the community structure within each microhabitat. And you can see here in the looping, you can see these are the different trophic groups of nematodes. And this gives us the indication what is, if you have a high mineralization rates, if you have loss of nutrients. So in the looping habitat, you can see we have a much more higher contribution of bacteriivore nematodes, which indicates high mineralization. But at the same time, you might not need this mineralization of nutrients. They probably get lost. So this is an invasive species that comes in and changes the heathland. And the nematode shows that you have actually higher mineralization there.
Okay, and you can predict those distributions from
we are
simple
trying
NDVI?
to do it. Not yet. We have not tried. We are not happy with our models yet. So we need to work more there. This is not simply the NDVI. We need a bit more to work on that.
Okay.
But we did find a good relation to the abundance. So this gives us a starting point. But we are still work. So if anybody is interested to join, I will be very happy to include in the group.
Okay, thanks.
Sorry, Maria, just a small regard. Regarding those bacteriivores with lupine, you mentioned that lupine is a nitrogen fixating plant.
Yeah.
Well, actually, it has a lot of nitrogen fixating bacteria. So probably the diversity and the abundance of those bacteria cause the higher concentrations of bacteriivores inside soil with lupine. It
is very possible, yes, of course.
Yeah.
Because of the higher bacteria, yeah, of course.
As every leguminous plants, it has those bacteria, and it has those huge tubers where the bacteria that fixate nitrogen from
yes.
There. So that's probably the answer to that.
Yeah, yes. This is the cause, of course. This is the cause.
Okay. Thank you, Maria.
Thank you.
Thank you for the comments as well. So let's move to the next presentation. Is from Martin. Hi, Martin. Martin Mokroš.
Yes. Hi. Yeah. Can you hear me?
Okay.
Okay.
Okay. Martin will give a presentation on merging ground and air-based 3D points clouds to gather precise data about individual trees within dense forest. Is an ongoing research. Okay.
Okay, so can you see my presentation?
Yes, we see it.
Okay. Okay, so I have prepared a presentation about our ongoing project. And ongoing, it means that we are now in the field and doing all the data acquisition. We get two projects to reach the goal that we have established and we would like to solve. So this presentation will be about lots of hypotheses, hopes, and some kind of dreams to solve some issues that we have. So what we have in mind and what we want to solve and why. We have lots of not lots of, but we have some research from past that we have focused on different platforms for the forest. So for example, terrestrial laser scanning, also for the photogrammetry, mobile laser scanning, and also some kind of low-cost solutions. What is like something like smartphone or cheap cameras, and also we have some project that where we are using drones, also copters and fixed wings, and in my group we are focusing to implement different methodologies to use these technologies, but also we are developing some hardware, and also we are working on developing the software how to process the point clouds to get the information about the forest. And mainly it is the diameter of the trees, of the trunk, the height, the volume, and information about the crowns. And this is within the forest, mainly managed, and also we have some unmanaged forests. So these forests are closed, I mean canopy way, that we don't see anything from the drones. So we have this ongoing project focusing on these technologies, but separately. So every time we are finding that, okay, this kind of technology is really good to get this information, it is really precise, but there is lack of information of, for example, from terrestrial scanning, we have really good information about trunks, but the information about crowns or about the height is not so precise. But from the other point, from the UAV or from drones, we can see really good like crown characteristic, what is the diameter. Also, when we have LiDAR data from the airplane, we can also have really good height. So we have decided that we will focus on merging these platforms to have ground technologies and then merge them with UAV. We have really good results from terrestrial scanning, but also we want to use mobile. What is this one? Is handheld mobile scanning, so it is much more faster, but it's more noisy. We will also use new iPad Pro because they implemented LiDAR inside it. So the promo videos looks great. So if it's going to be 20% from that, it could be good. So these are like active sensors that are shooting beams. So it is more precise, we can say, in some cases. And we will use also passive sensors for these images. So photogrammetry, so also mobile and static photogrammetry, and also multi-camera system that I'm currently developing. And we want to also implement smartphones to see if we are able to capture the three-dimensional forest, and it would be really beneficial to use phones from the practical way. So everybody has smartphone also from the foresters, and we can use also GPS. So we will also look on the accuracy of GPS under the canopy. So you can see these are some results that we have. So you can see the noise is much lower than passive sensor. So is the result from the terrestrial scanning on the right, example from photogrammetry from images. So why not to use just TLS? Because we have to also look on the price, and the photogrammetry is much more cheaper from the point of hardware. and also can bring some other things, like for the visualization, if we took the images properly, the visualization is much better with photogrammetry. And also we have now ongoing project that will also implement deep learning with images. So this could be really beneficial for the photogrammetry when we will implement deep learning to do the tree classification from images. So this is just one example from previous paper that we focus on the different data acquisition and compare what is good and what is not very good. We find out that it is quite different also data acquisition when we compare the laser scanning and the photogrammetry. And but we have this linked. So right, we will establish plots and we will do all the acquisition on the same plots. And we have already started to plot, and we will use all this equipment to do that data acquisition and compare everything together and find out what advantage we can have from each device. And then we will also work to our software because the noise is different, the point will be different from equipment, so we will do modification to get the best accuracy from each. So here is some example. If you can take 20 images around, and you can really accuracy. This from the project that have researched. We detect the annual movement of the annual and this and to catch that with the return. It is really good proper. And this is something that I have worked the projects, and this is the main field of multi-camera system. So I have different prototypes that I want to compare with all those devices I have mentioned. On the left is one prototype that is for the forest road. In this like low-cost multi-camera system that requires and cameras. So for a multiple camera system, I don't know, 200 €. And on the right is first test of this handheld mobile photogrammetry prototype. But why the multi-camera system? It's there is more reasons why to do such thing. You need to scale the data, and if you have multi-scale the data, the position of the camera. And the other thing is that when you are using Canon, it's rotating around, so you have 360 movement, and you can see everything around on the top and everywhere. But with the camera is different, so you have to move with this one camera, and it is really difficult to do such capturing, to also capture the ground one camera. So this is the why to do such thing. This is the prototype, is also the proof. And the drones. Now when we have used, we have the research about the RTK and PPK, so there is a typo, sorry, in the forest, and it is surprisingly accurate. So you don't need to use ground control points. So within our forest, it's also impossible in some places because you don't see any ground, so you cannot put ground control points on crown. It's not possible. So with this solution, now we are able to do really great things with drones in dense forests. So we are using a quadcopter, also fixed wing, and with quadcopter we have really high resolution, but the plot is much smaller. And with fixed wing, we can capture from one flight 300 hectares, and the accuracy and also resolution is really good. So we have flow between, I think it's like 300 hectares. So these are just examples. And we have also the capture during the leaf on and leaf off to reach the line, get the ground. So we have a fixed 3D camera that is doing occlusion in one, and then we are able to capture the ground just with the ground leaf season. When we match it on from right side the accurate really super just on the photo information. We have established and we will find the best solution for us. This is example from just one part from video. And this is the count from the leaf off and on leaf on. Okay, and that's it. Thank you very much.
Thank you, Martin.
Okay, I was wondering during the presentation that if somebody can hear me and see me, so it was okay.
Yes, we can hear you clearly.
Okay. Don't worry.
So, do you have questions? No?
So yeah,
Okay, thank you.
have one short. So basically what you're saying is that in deciduous forest at least, you can achieve high enough accuracy and resolution with just using photogrammetry, so you don't need basically any active sensors.
Yeah. This is something I think really interesting, and I didn't see it. I think it's also possible because we are using this RTK, it's really accurate. So you can merge the data from different seasons and it will be really accurate. You don't, because sometimes with ground control points you have lots of issues. The accuracy sometimes is not very good. And also there is some kind of new approach with fixed wing and these 3D cameras that in one fly the camera is moving and you are getting these oblique images and other images, and it's surprisingly really accurate. And we don't see enough trees during this leaf off season, but we have lots of points on the ground.
Cool, really nice.
Martin, do you plan to use small laser scanners carried on a UAV to measure forested areas? I mean, there are some small size laser scanners on the market already, like Velodyne products.
Currently we are doing like a review paper with guys from Finnish Geospatial Research Institute, and we have also done this review on these LiDAR sensors for UAV. So I need project to implement something like that, because it's cheap, but you have to have lots of things to put it together. We are now have discussion with a colleague to use a helicopter for this area with LiDAR, but we will see if this going to happen. I don't know, but this is like a VIP solution, like this low cost thing. We will see. Yeah. But I would like to do it.
Okay, thanks.
Okay, thank you very much, Martin.
Thanks.
So let's move to next presentation that is from Paulus Gernot. Paulus there? Hello, Paulus?
Okay, hello. Can you hear me?
Yes, we can hear you. So Paulus from Carinthia University of Applied Science in Austria. And the speech is on geoinformation meets art in the forest at the football stadium in Glasgow, Austria. Okay, we can see your first slide.
Oh, can you see it? Okay, perfect. So hopefully this will also work. Okay, hello. Hello. I would also just like to give an short overview about the first project. Antonino was just mentioning about a very unique project about how actually vegetation monitoring has vegetation with art. So this would be the first one, and the second one is just finished, where we actually used to do photogrammetric modeling of grapes and try to derive here parameters for yield predictions. Work will be presented at the virtual conference at the University of Salzburg at the beginning of July. So I would just like to start now with this first project, which is actually really very, very special and unique. So the idea to this project actually goes back 70s to a black and white thing of an Austrian artist, Max Paintner, with the name The Unending Attraction of Nature. If you remember, at this time we had a lot of issues. This was the first issues of acid rain and massive forest destruction here, and he actually had this painting, and this painting was a year to come up with, to bring this painting actually to the real world.
Yes,
what you can see here.
Not see next slide. See the first one.
Okay. Okay. So
can you stop and share again?
Yeah, I will.
And maybe turn off your camera.
Yeah, I will
just
try
to reduce the traffic.
Let's see how this will turn off the camera.
Okay, Antonino, there is clearly some problem. Now I don't know. Can we move on eventually? And while if Gernot is not able to solve the issue,
I have also some problem with the recording, so I was taking advantage of this this stop to solve my problem.
Okay.
Okay, we can would like to move to the next one. The next one is a speech from Felix. Okay, Felix.
Yes, I'm here.
Frances, that is the working group vice leader, is the University of Politecnica of Valencia in Spain, and his speech on the benefit of remote sensing and the calibration of the eco-hydrological model. I would like to ask if someone can also record this part. Salvatore, can you record this speech?
I'm trying, but I don't have the rights. I think it's
Okay,
only you that are able to record. There might be some problem of the size of the files.
Okay, Fulvio is helping as well, so no problem. We can go forward. Thank you, Fulvio. Okay, so let's continue.
Can you see my presentation? No.
Yes, we see your first slide.
Oh, okay.
No, your second slide. Number 7.
This one, the first one. Yep.
Okay.
The first one. Okay, well, the idea is to cheer you up because all your work can be profitable for eco-hydrological modeling. That's the idea. But in this case, I'm using satellite information. But at the end, the idea is the same: the use of remote sensing to improve our modeling performance. The general question is, it is possible and profitable to use remote sensing information for model calibration. But why remote sensing? Because the main advantage is that remote sensing is giving us a spatial coverage of state variables different than the traditional discharge, the Q, which is only an aggregation of the hydrological processes at some points in the catchment. Remote sensing is giving you the spatial, or can give you the spatial variability of some of the hydrological or eco-hydrological processes. The only disadvantage is that in theory we have higher uncertainty than field observations. And why we need to calibrate our mathematical models, because any mathematical model deals with effective parameters different than even the unknown true values of all parameters for different reasons, because errors in the information, because the spatial and temporal scale effect in the numerical integration, etc. So that's the reason, because with calibration, the model performance usually increase. The first thing to make this possible is to develop and apply a spatial temporal objective function which can capture the spatial pattern, the temporal and spatial pattern of the remote sensing information on one hand, and to evaluate the probability. What we're going to do is to use, in one case study, the surface soil moisture from satellite as observed state variable, and to compute the performance comparing the use of this surface soil moisture. We will call it configuration 4. I'm going to present only one remote sensing configuration against the use of just the discharge, I will call it configuration 1. Also, for validation, I'm going to present you the comparison between the leaf area index simulated and observed from satellite. We are using our eco-hydrological model called TETIS. It's conceptual, but very important with physically based parameters, which make easier the estimation of the parameters from a practical point of view. We are trying to reproduce all important process, but always trying to use the minimum number of parameters. In fact, we usually for hydrological modeling, we use just 9 parameters, 8 for production and 1 correction for the propagation in the river network. Also, that is an integral model, means that we use the same model for water resources, floods, sediment cycle, erosion problems, dynamic vegetation, as in this case, crop production, nitrogen and carbon cycle, etc. Reservoir, snow. But the important thing to include the remote sensing information is that the specific state variable that you are observing is modeled by your model, in this case, the surface soil moisture. In a space is distributed, it means that it will reproduce the spatial variability of the hydrological cycle using all the spatial information available, reduce the spatial scale effect because the size of your control volume to do the numerical integration is smaller. And from a practical point of view, it's very interesting, believe me, that it gives you results at any point in your catchment. Internally, the effective parameter is split in 2 components. On one hand, the value estimated at each pixel plus or multiplied by a correction factor. So what we calibrate is not the value of the parameter at each pixel. What we calibrate is this correction, which is the hypothesis is that the correction factor is common for all pixels, but different for each parameter, of course. In this way, we maintain the spatial pattern of the initial estimation of the parameter maps, and also it is possible to use automatic calibration using relatively simple optimization algorithm. For the dynamic vegetation component, we use the state variables, the leaf biomass, and the dynamic equation is based on the light use efficiency here. Well, the idea is this dynamic equation is connected with the water cycle through a stress factor, which includes the water stress, and the transpiration is a function of this stress factor and the leaf area index. So the leaf area index is also the state variable in our model, and we can compare the observation with the simulation. At the end, for vegetation, we have 11 parameters for each type of functional type of vegetation, but in practice, the influent parameters is much, the number of influent parameters is much smaller than 11. Just for the sake of simplicity, for temporal efficiency, I'm going to show the results using the Nasuclive efficiency index, and also I'm going to use this efficiency index for the optimization in configuration 1. Probably you know perfectly what is this index. And the most interesting part is how to include, how to measure the similarity between observation and simulation in the case of spatial state variables. In this case, we use the principal components analysis, and we built this index as the sum, a weighted sum of the Nasuclive efficiency index of the loadings for each of the different principal components. And the weight is the portion of explained variance. Well, the idea is that this metric of any other metric should incorporate the spatial pattern for sure, and the temporal dynamics without considering the exact values of the satellite or your VAD. If we are feeling is that the exact value is not is different from the observation. The case studies relatively large catchment in the east coast of Spain, but is with a Mediterranean semi-arid climate. Mediterranean means that there is clear bimodal precipitation in spring and in autumn, and the potential of evapotranspiration is much higher than, in this case, just higher than the annual rainfall. Most of the catchment is natural, with pine forest, but this semi-arid climate plus karstic lithology produced that this 1,500 square kilometers catchment is an ephemeral rhythm. For observed state variables, we use, in the case of the surface soil moisture, the product from Barcelona Expert Center. In the case of lye, we use the product from the models from NASA. The daily discharge was from a Spanish database that you can access from the web directly for daily time step. We have a common observation between 2010 and 2015. Then we use 3 years previously to this common period for one up of the state variables to be sure that everything is not affected by the initial condition, and we use 3 years for calibration and 2 years for validation. At the end, what we calibrate are only seven correction factors for hydrology, because this is ephemeral river. Then the groundwater is not working, of course. And concerning vegetation, actually only three, in this case, three parameters or influent times 5 main natural land covers. Then we have 7 plus 15 variables to be calibrated. In configuration 1, we use just the flow at the catchment outlet, and the objective function is the NAS supply efficiency index. So the calibration is relatively good, NAS index of 0.9. In case of ephemeral river, actually it's more important to have the probability of zero discharge clearly simulated, which is the case. But when we go to validation, there is a strong deterioration of the NAS from 0.9 to 0.46. We look at the mean areal soil moisture in the upper panel and the mean areal, the areal mean leaf area index. Through time, we see that, well, the soil moisture is relatively good simulated, and actually the leaf area index has a negative NAS supply, but it's just because this bias. Look at the scale, actually it's a small bias. The reproduction is relatively good. It can be impossible from the discharge to correct this bias in the leaf area index, because there is not transfer of, there is not information in the discharge that can correct this error. But the problem is that when we look at the spatial simulation in the calibration period and in the validation period, it's a disaster. This is for the soil moisture. There is no, the determination coefficient are always smaller than 0.4. And very often around 0 actually. It's like a random simulation in a space. The areal mean is okay, but the distribution in space is almost random. And the same if we compare with the leaf area index. Of course, it's worse because we didn't use the leaf area index is more far away from the information provided by the discharge at the outlet. What happened if we use just the remote sense surface soil moisture? Well, in this case, we use as objective function our spatial temporal efficiency index. The calibration is relatively good, 0.6, concerning the mean areal, the areal mean temporal evolution of the soil moisture during the calibration. But in the validation also is relatively good, with a very small degradation of the performance, which is something important in mathematical modeling. And if we look other things, if we look the discharge, we didn't use the discharge for calibration. This is the validation. The validation is almost similar than using the discharge for calibration. And the leaf area index, in this case, the bias was corrected and given relatively good value. and we look at in a space, then we can see that the coefficient of determination, the R-squared, is almost positive in all pixels. I would say in all pixels actually, for the surface soil moisture, and a little bit worse concerning the leaf area index, because we didn't use the leaf area index in the calculation. So going to conclusion, the first day is that it's possible to exploit the remote sensing data to calibrate satisfactorily a distributed eco-hydrological model that engages basins, which is the configuration 4 situation, because there is a transfer of variable information from remote sensing observed state variable to most of the eco-hydrological processes. Of course, there is no transfer of information from surface soil moisture to groundwater permeability or to river cross sections, but concerning the upper part of the soil, there is this transfer of information, which is affecting even the discharge of the outlet at daily time step. And which is for me more important, the use of remote sensing, or as important as the previous conclusion, the use of remote sensing produces a more robust model with less degradation between calibration and validation and with a spatial coherence, not achieved using only the discharge at the outlet of the gas. And that's all. Thank you for your attention.
Okay, thank you, Felix. Thank you. I have a question for you. In your opinion, is more important to have remote sensing images with high frequency, temporal frequency, or do you prefer to have higher spatial resolution?
In Valencia, we say with more sugar, more sweet. If you have only one picture every month, we can use one picture every month.
Because you know there is a balance between the temporal and the spatial resolution. No? If you have very images with the high temporal resolution, usually these are with the low spatial resolution, and vice versa. If you need the high spatial resolution, and then you have low temporal resolution.
Well, then it will depend of the spatial and temporal variability of the state variable measuring. Then for vegetation, maybe you don't need a higher resolution in time, even in space, it will depend. But on the other hand, for surface soil moisture, you will need, it will be better to have a higher temporal
resolution.
At least weekly. I would say for surface soil moisture, you must have weekly, one per week. For vegetation, with one per month, you can have valuable information there.
Okay. There is a question from Salvatore Manfreda.
Hi, Felix. Thank you for the presentation. Just a quick question. Did you explore different data set, different satellite data set at different resolution, and to explore applicability of US data?
For the second question, yes. The idea is to see how to incorporate UAV remote sensing information here. There are, in my opinion, there are two possibilities, or to improve the reliability of the satellites, because the problem with UAV is your spatial coverage. You cannot deal with 1,500 square kilometers of this catchment, or even it will be difficult for 100 square kilometers. But you have, for sure, more precise information in a space on one hand, and I think with less uncertainty of the values that you are observing. So you can merge these two types of information. That's the best way. You're thinking in practical eco-hydrology with medium-sized or large catchment. For experimental basin, of course, you can use directly the UAV, or for an experimental plot, you can use directly the remote sensing information. That can be a first approach. And the idea, yes, is to check different resolution, including UAV data.
All right, thank you. Thank you so much.
Thank you, Felix. So we are a bit late, but I would like to give again the word to Gernot.
Okay. Thank you.
So let's try again. Maybe.
Okay. Yeah, I hope
if you can have a 10 minutes presentation.
My connection will work. So.
Okay.
So can you see it now?
Now, yes.
Okay, so hopefully this will work. Yeah. So as I said, this was actually the idea to bring this picture to the real world. And so, do you see now also the next slide?
No.
I see always the same slide.
Okay.
The vision of Max paint man.
Okay. So I will just maybe go inside the PowerPoint. Is it now working?
Okay. Now it's changing.
Okay. Okay. So what you can see here actually the real world implementation. This was done by Swiss artist Littmann, and what he did actually, he brought this idea in the 70s of Max Paintner to the real world, and this was actually the largest public temporary art installation in Austria. And what he did was that he planted a European mixed forest of about 300 trees into a football stadium in Klagenfurt in Austria. This was one of the stadiums when the European Championships has been played. And the idea was now actually to use this art installation, the forestry experimental laboratory, to use different remote sensing here, the whole area, and also the forest. So this is actually a little bit similar to the work Martin was presenting. So it was to create the full 3D model, the digital representation of this temporary art installation. Installation was in last September for about 6 weeks. And if we go now here to the, hopefully to the what we used here was a combination on the one hand of photogrammetry, using an amateur system to get photogrammetric, to generate a photogrammetric point cloud. Here you can see, and the mission plan. And on the other hand, also a terrestrial laser scanner. This was an RS 360 from Geosystems, where in total 560 lens has been actually used inside and outside the stadium, really to collect also the complete listing from the ground of the forest. And what we also did was a full forest inventory, where we actually mapped all the tree species and also measured the breast height diameter. So just to show you here also then some of the first here, on the one hand the auto image with a resolution of mosaic with a resolution of 6 millimeter. Then you can also see here the photogrammetric point cloud, which has about more than 800 million points, just the forest itself. And what you can see here now is the fused point cloud from the stadium using the terrestrial laser scanning, and also the photogrammetric point cloud. So we also then some comparisons. What you can see here in this red and green image is one hand in red are all the dots which are from the rest where you can get the information from this side, and then on top where you do not have that much information from the terrestrial laser scan, green are the points from the UV point cloud. And also the cloud-to-cloud registration here worked here very well. So we have an error of it was possible to match the point clouds with a centimeter resolution. And here you can also see then the differences of the point cloud. This is here this iterative closest point distance here in red. In blue, we can see that where we do have the information of both here, also we were able to achieve a quite nice match. And some other first results here was that we generated here a complete revision plan where each tree is located together with the information about the species, and also derived then here some canopy height model. And here we are still in the process now to actually do some further assessment and comparison of the results. But this is actually was a pretty unique project. So maybe if you have heard about this also here in the media. And the second project we just actually finished was also to use to try to generate a full 3D model and also try to classify the grapes using the point cloud. So at the point cloud level, in order to determine here some morphological parameters of the grapes, which in the future can be used here then also as one component for yield prediction. We used a bunch of grapes in a lab setting. Here you can see the image. Then we have here the 3D model, and based on this 3D model here, we were actually also then able to identify individual 3D objects at the single grape level. So here are also 2 videos behind. So maybe I can just, I'm not sure if you can see now that actually the model is turning here in the middle, and I think we also have here the same here at the 3D of this, and then also this was one work of one of our master students here. He actually took then a lot of reference measurements in determining here the volume, width, length, and also the weight of the individual grapes. And just to show you here then some of these comparison was that actually this worked out quite nicely. So we were able actually to identify the correct number of the grapes compared from the real grapes to the 3D models here. And also the volumes fit quite well together. There were some problems here in terms of matching between the green grapes and the red grapes. So with the green grapes, it worked much better. And we are also actually we are able to show now this is actually also working here, at least at the lab scale. So in the next step, we are also planning to take this out to the field and see how we will be able actually also to measure these morphological parameters here then out in the field at the vineyard scale. Okay, so that's it. Just a brief and short, and so I hope this has worked now so far. Thank you.
Thank you, Paulus. Thank you. So, any question from the audience? No? Okay, thank you, Paulus. So we can go to the final remarks from Salvatore in order to end this nice session we have been, right now we are 27 people, we have been even more during the day. So Salvatore would like to end this session.
Okay. Let me check if I can. I have prepared a small presentation as well. So I agree that this has been a great initiative. I would like to thank you, Antonino, for organizing this. I would like to thank you all presenters for their help and support. Can you see my screen?
Yes, we can see it.
Okay, and of course, as a final remark, I would say that this cost action is now forced to move part of its activities on the web, and we're doing our best to keep alive the networking among all scientists. And this network of scientists is growing with time in a significant way. The number of participants that we had during the morning somehow testifies that we are a good group, because the working group 2 has been able to collect more than 33, 35 participants during the day. Of the action is somehow to exploit the use of UAS for environmental monitoring, and we are now merging together knowledge and information available from remote sensing and try to enhance remote sensing using UAS. You will see the cost action is one of the largest cost action that is running under the cost association. The structure of the cost action harmonious is given in this slide, and I will upload and we will say in the memory of this meeting for those that are interested. And you can contact the leaders if you want to be involved in one of these working groups. You can follow our initiatives through following one of our channels. We have different channels that are listed here, our website, and also our socials and YouTube channel. On YouTube, you will find a video of this meeting of today. And just a final remark is that regarding one of our last publication, that somehow synthesize the aim of the action, that is try to identify guidelines in US applications. In this context, with Goran Tumsic, we published a paper just a few months ago on remote sensing, and this paper addressed the complexity of US observation and the number of skills and knowledge that is needed in order to address properly a US survey. This complexity, we try to reorganize the workflow and the procedure needed in order to obtain the best result in US-based mapping. And we divided the workflow in 5 different steps. And within these 5 different steps, we identified a general workflow that you can revise and check on the manuscript. Just as a final closing remark, and a project that is running, that is for me very interesting and stimulating, is the questionnaire that we are running. We are trying to make a general survey among scientists and among researchers in order to identify what are the main challenges and open questions in drone-based studies. And if you are able and if you have time, it would be very great if you can contribute to this survey. Here you can find the link. Jana already introduced some results, and I have to say that while we were talking, some of you probably already participated to the survey. We have small discrepancy in the female participants. So if there are females in the audience, please contribute and support the survey. There's also quite heterogeneous participation in based on the position of each of us. And in this, we see a lack of participations in people from private sector, and this is obviously due to the fact that most of us are coming from research and academia. And finally, there is also a relative variety of interests and in specific landscape with a prevalence of natural and protected ecosystems among the others, but also many people is interested in agriculture and precision agriculture. So closing closing the presentation, I would say that we are an open action. The aim of the action is to promote the use of drones for environmental studies and in order to monitor environmental system, in order to enhance our capacity to predict water resources availability and assist agricultural ecosystem management. And we have already gained some results in the definition of standardized protocols that will be further investigated and detailed in the coming year in order to specialize these outcomes in a specific context related to each of the working groups, meaning looking at the vegetation monitoring, looking at the soil moisture monitoring, and river monitoring. This is pretty much with this. Let's move to different contexts. This is pretty much what I have to say, and in a general context, I have to say that there is a wide number of participants that are effectively contributing to the activities of the action, and I think that we will more or less in a year from now should be able to finalize the protocols for vegetation monitoring, for instance. So with this, I would like to thank you all, and probably Antonino, you can say a few words to close the session.
Okay, thank you, Salvatore. I just want to remark if there is any one of you that is not yet within the KAUST action and would like to join our research group, is a great opportunity to make networks of people that often have similar objectives and study topics. So it is a great opportunity to make a network of researchers. So if you would like to join the working group, please write an email to me, antonino.maltese@unipa.it, or if you would like to join some other research group, write the action chair, Salvatore Manfreda, or the other working group leaders. And I hope to have the opportunity to see you again in person in next future. Hopefully things are going to hopefully are going to change, and we will have less travel limitation. Thank you very much for attending. I very appreciate the number of people that attended the today meeting. So thank you very much to all of you and to the speakers. Thank you.
Bye.
Thank you, Antonino. Bye-bye.
Ciao. Ciao, ciao.
Okay, thank you. Goodbye, everyone. Thank you so much.
You.
Have a nice day.
Bye, everybody. Ciao, ciao. Ciao.
Ciao.
Arrivederci.